In a quest to look for specifically immune biomarker, and the most studied immune biomarker currently, PD-L1, specifically for TKIs and for cabozantinib, we looked at PD-L1 status on the CABOSUN trial, the trial that led to the approval of cabozantinib versus sunitinib frontline, and on METEOR trial, the trial that led to the approval of cabozantinib second line or later post-TKI. That trial was against everolimus. And when we looked at PD-L1 immunohistochemistry, we looked at different cutoff, 1 and 5%. We looked at PD-L1 immune density. We looked at PD-L1 on tumor cell, immune cells, several lines of PD-L1, what defines PD-L1 positivity. And what we found is, irrespective of the treatment, PD-L1 positivity is associated with worse outcome, worse progression-free survival, and worse overall survival. Nevertheless, compared to the control arm, sunitinib on CABOSUN, everolimus on METEOR, cabozantinib does better. And I think this will be important in a way to define what are the benchmark for PD-L1 positivity in terms of clinical activity, PFS and OS, especially when we start combining agent with cabozantinib. There is one ongoing trial combining cabozantinib with nivolumab, and potentially other trials are in the making. One led by Dr. Pal is combining cabozantinib with atezolizumab, a smaller study. So this, I think, will be important for the future for combination to try to benchmark in PD-L1 positive patients.
Okay. And if PD-1 is associated with the worst result for cabozantinib, is there some kind of synergy you're hoping for with the nivolumab, atezolizumab arms, or could it just end up making things a lot worse a lot quicker?
Yeah, it is. I mean, this is an interesting question. It's possible that it's simply a PD-L1 positive patient do worse. Though they may do better with checkpoint blockers, they do worse in terms of everything else. We know very well that in localized disease, PD-L1 positive patients have a shorter recurrence-free survival and overall survival. We know very well from looking at the COMPARS study with Dr. Moser and Signoretti that if you get frontline sunitinib or pazopanib, if you're PD-L1 positive, your overall survival and progression-free survival is worse. So I'm not surprised that PD-L1 positive patients do fare worse on sunitinib, everolimus, and cabozantinib. And I'm also not surprised that cabozantinib maintain its efficacy over sunitinib and everolimus in CABOSUN and METEOR trials.
Well, with cabozantinib established as superior to sunitinib, that leads to the JAVELIN trial in which sunitinib was the control arm. Was that just due to the timing of trial design? Absolutely.
At that time, it was due to trial design, and there are still ongoing studies on with sunitinib as a comparator arm, which is a less than appropriate control at this point. Perhaps not so much in the good risk, but in the intermediate and poor risk. Nevertheless, that study enrolled and finished accrual some time ago, and they clearly showed superiority with the combination over sunitinib. Response rate, progression-free survival, both in the PD-L1 positive patient and in the overall population. Overall survival, we're eagerly awaiting that. Hopefully we can report on that soon, but there weren't enough events to conduct really the analysis with much precision. The follow-up for survival was less than a year on the study.
So do you see perhaps a future in which a patient's PD-1 positivity sorts them to either the JAVELIN trial or cabozantinib, or is there any future for a headline comparison between those two as well?
It's possible, although when you combine a drug, especially PD-1, PD-L1 with a VEGF TKI, I wonder if they do worse with a VEGF TKI, but then they do better with a PD-L1, PD-1, if then at that time you do not need the biomarker. And fair enough, at least if you look at progression-free survival, it was the same in the whole population versus the one that is PD-L1 positive. So we may not actually need the biomarker at all. Interesting thing about the JAVELIN study too is that on subgroup analysis, looking at the IMDC or the MSKCC risk group, there was benefit in all three risk groups. While in the 214 trial, the nivo-ipi trial that led to the approval of the combo of nivo-ipi versus sunitinib in poor and intermediate risk, the benefit of sunitinib in terms of response rate and PFS at least outweigh nivolumab, ipilimumab in the original publications.
Okay. That sounds like a very interesting and complicated time working in renal cell carcinoma then.
I think so. I think we're going to have a lot of first-line option, at least in the United States, approved, or at least be able to use nivolumab, ipilimumab, avelumab, pembro potentially, cabo-nivo, levantinib, pembrolizumab. Also, let's not forget atezolizumab. That study showed that that regimen is actually pretty well tolerated, and the study met its primary endpoint of progression-free survival in PD-L1 positive. So to me, the unmet clinical need and the big question, what to do second line, and can you still use these PD-1, PD-L1 inhibitor after progression on first PD-1, PD-L1 inhibitor. So in the past, we have used TKI after TKI, but these were different TKI. We've never used almost, let's say in RCC, everolimus after temsirolimus or temsirolimus after everolimus. How different they are. Can you put a patient, let's say, who progressed on avelumab, can you put them on single agent pembrolizumab or on pembro-axi or pembro-levantinib or atezo-bev? Do you need to put them, or can you just use a plain TKI? Totally unmet need, very important questions, and hopefully the next few years we'll be able to answer that.